So welcome everyone, and thank you for joining us for today's session as part of the CPHI webinar series. Today, we are presenting the necessity of extractables and leachable qualification for lyophilized drug products in a session sponsored by Nelson Labs. My name is Ana Castro, and I am a senior content producer at Inform Markets, and I'll be moderating this session today. Just a little housekeeping before we get started, remind you that this is a live session. So if you have any question during the presentation, please type them into the question box in your control panel, and please feel free to do so throughout the session, and I'll try to put as many questions as possible to our speaker at the end. Also let you know that our speaker today is happy to share the slides of his presentation, and if you wish to download them, you can do so within the related content tab that you will find on your panel. So here's today's agenda, and this webinar will present you a true understanding of the interaction mechanism between a lyophilized drug product and its container, as well as provide you with an understanding of an extractable and leachable study design for a lyophilized drug product. So without further ado, I'd now like to introduce our presenter for today, Pete Christians, Scientific Director at Nelson Labs. Since 2001, Pete has held his position where he develops analytical methods and protocols for both extractables and leachable studies for the pharmaceutical and medical industries. Pete oversees all laboratory operation at Nelson Labs Europe, and it's also giving support to business development and R&D. Now, I'd like to hand the session over to you, Pete.
Well, thank you very much for the introduction, Diana. So first of all, I would like to thank you all for attending this webinar. I would like to thank CPHI for the invitation and the organization of this event. My name is Pete Christians, like Diana said, and I'm Scientific Director of Nelson Labs Europe, which is a CRO that is specialized in performing extractables and leachable studies for a wide variety of drug products as well as for medical devices. And today I will talk about the necessity of extractable and leachable qualifications for primary packaging of lyophilized drug products. And I will try to explain a little bit more in detail why there is such a debate around the depth of extractable and leachable qualifications. And that will also be based upon our experience when we have performed historically these kinds of extractable and leachable evaluations on the container closure systems of lyophilized drug products. I will also try to address some of the fallacies that exist, or misconceptions that exist in extractable and leachable qualifications for these kinds of drug products. Oh, yeah. So I'll immediately go to the table of content. So in the first part, we will have a look at the primary packaging containers for freeze-dried or lyophilized drug products, as well as some of the most common administration devices or drug delivery devices that are used for reconstitution or after reconstitution. In the second part, we will have a look at the US guidances and European guideline and what kind of information that they give us in terms of depth that is needed for the qualification of the primary packaging of the lyophilized drug products. In part 3, we will have a look at the general flow of an extractable and leachable evaluation, more in general terms, you know, when you are looking at the pharmaceutical primary packaging in combination with a drug product. Part 4 will go a little bit deeper into the interaction mechanism between the lyophilized drug product and the materials of construction of the primary packaging. And I will try to explain that the mechanism of interaction is not based on a solid interaction between the rubber and the lyophilized drug product, but it's more some kind of outgassing mechanism. In part 5, we will have a look at what kind of consequences that has in establishing or designing the leachable studies for lyophilized drug products. Part 6 will go a little bit deeper into the reactive leachables, something that we often see in these kinds of applications. And in the last part, we will have a look on how to minimize the interaction between a lyo cake and the stopper. But let's first have a look at what kind of devices we are talking about. When we're looking at the types of containers that are potentially maybe in contact with the lyophilized drug product, or we can subdivide it into 3 categories. The first one is the primary packaging that is the long-term storage of the lyo cake. Second type of container is the container for the reconstitution solution, which is typically a vial or a prefilled syringe. And then you have the drug administration sets or the drug delivery devices that will be used to administer the drug to the patient. When we're looking at the container closure system for the primary packaging of the lyo cake, it is typically a vial with a rubber stopper. It can also be a dual chamber vial, where in one chamber the lyo cake is contained and in the other the reconstitution solution is contained, or a dual chamber prefilled syringe is also possible. So the rubbers there, whether it is a stopper or a plunger, are selected based upon their low residual moisture content and the low water vapor transmission, because of course you want to keep the lyophilized drug product as dry as possible, otherwise it could affect the stability of the drug product. A second type of container is the one that contains the reconstitution solution. Like I said, it may be a vial with a reconstitution solution or a prefilled syringe. There, what you actually need to do in terms of E&L qualification is exactly the same as you would do as for an lyo cake. So you need to perform an extraction study and a leachable study. And the third group of devices that will be in contact with the drug product or the drug administration sets, such as a disposable syringe, an IV bag, or a pump system with the administration tubing. We will not go into detail on the last category, because that falls outside of the scope of this presentation. So let us look at the US and European guidelines, and let us first look at what the US guidance is for these type of drug products. Here I would like to refer to the table that ranks the packaging concerns based upon the likelihood of interaction between the packaging materials and the drug product, and also the degree of concern associated with the route of administration. This table first appeared in the FDA guidance container closure systems for packaging human drugs and biologics, and then was also included into the USP 1664 in a slightly modified way. But what does it actually mean for small volume parenterals in general, and also for the lyo applications? So what is the difference between the 99 guidance of the FDA and the USP? So first of all, the degree of concern associated with the route of administration stays the same. It is the highest concern because you take away a very important barrier, which is the skin, and all leachables or impurities will be administered to the patient, and they will be circulated through the blood and may cause potentially systemic adverse effects. However, what we have seen here is that from going to the 99 guidance to the USP 1664 is that the risk of the interaction between the drug product and the components has been reduced. So it went from high to medium risk. The reason for that is that most of the parenteral drug applications are aqueous-based, and there you do not expect too much interaction between the drug product and the components. However, you have those drug products that have a higher organic load, or you have sometimes drug products that are not aqueous-based, so there the risk remains high. Now, what does it mean for lyophilized drug products? So you can see that we're looking at the sterile powders and powders for injection. There, the risk has been reduced from medium to low with the logic that there is only a very limited potential interaction between the rubber stopper and the lyo cake, because there's no direct contact in a lot of cases. However, we will see later on that the mechanism of interaction is not based upon a solid-solid. interaction, but based upon some kind of outgassing mechanism. So to conclude on this, we can say that the low likelihood of packaging drug product interaction for small volume parenteral application is based upon the observation that the interaction between the solid and the material is limited, and that there's a limited direct contact between the lyoquake and the rubber closure. However, the interaction mechanism between the lyoquake and the components is not based upon direct contact, but based upon an outgassing mechanism. So we have to be careful in rationalizing the lyo applications as being non-critical. Now the situation in Europe. Here I would like to refer to the EMA guideline of 2005, the plastic immediate packaging materials. It contains some kind of decision tree on what to do for certain dosage forms. And for the parenteral solid dosage forms, the conclusion is that you only need to give some specification on the materials and perform an interaction study if necessary. So in the text, they refer to what is stated here. So for solid active substances and solid dosage forms, the risk of interaction is low and generally does not require a container content interaction study. Solid dosage forms intended for inhalation or parenteral use, however, like lyophilized drug products, may need interaction studies and so on if necessary. So here, the text could be interpreted as ENL studies for lyo products are more optional, but in reality that is not the case, because the European authorities always will request a full in-depth ENL evaluation as for any other parenteral application. So the conclusion is there's some potential ambiguity in the regulations in the need and the subsequent depth for ENL evaluations for lyo containers. There's a difference between the written regulations and the practice or the expectation from the authorities. So how does an extractable and leachable assessment looks like in general? We're not talking specifically here on, about lyophilized drug products, but more, for instance, for the general category of small volume parenterals, for instance. In a first step, so after a careful evaluation of your materials of construction, you first design some kind of extraction study that is based upon what you know about the components and the materials of construction and the physical chemical properties and the composition of the drug product. This may be reflected in the experimental conditions, such as the selection of your solvents. pH can play a role, the time and temperature during the extraction, or the stoichiometry, for instance. This is something that you can find in the USP 1663 on how to design your extraction studies. And typically, when you generated the extract, you analyze those extracts with a series of combined methodologies and analytical techniques to fully characterize the extracts and to develop some kind of extractable profile of the materials. Once that has been done, you need to look, typically you end up after the extraction study at a number of 20 to 100 compounds. And then in the second phase, you need to say, okay, which ones here are really of risk? So you do some kind of initial risk assessment, toxicological assessment that doesn't need to be in full depth. For instance, you can use structure activity relationship assessments to do that. You also look at the concentration and you try to assess which of the compounds, of the 20 to 100 compounds, will potentially be at risk for the patient. And those ones you try to monitor in your subsequent leachable study with more quantitative or fully developed analytical methods. And typically, after a leachable study, you end up with a handful of compounds that you need to further assess for their toxicity and their dosing to the patient. And the best outcome, of course, is that after a full in-depth toxicological assessment, that no compounds are of concern for your application. Now, looking at the interaction mechanism between the lyo drug product and the primary packaging. So, like I said, it's based upon outgassing. And that is one of the most important fallacies or misconceptions that exist for lyo applications. So you can see in this picture here that the lyo cake that is stored in an upright position does not have any direct contact with the rubber closure. So you would, if you would say, okay, the interaction only could take place when there's a direct solid-solid interaction between the lyo cake and the rubber, you could conclude that there's no risk here. But what is actually going on is that the rubber contains a lot of volatile and semi-volatile compounds because of the production of the rubber, for instance, solvent residues or oligomers or degradation compounds of the antioxidants, which are relatively volatile. They may start to evaporate from the rubber closure, and they float around or fly around in the vial. And what you'll see is that here, that eventually everything will be adsorbed onto the lyo cake. The lyo cake is a very good adsorbent because it's extremely dry and it has a very high surface area. So everything that is released in terms of evaporation or outgassing from the rubber will be concentrated onto the lyo cake. With this slide, it's a bit similar as the previous slide. We are zooming in on the rubber and the lyo cake. Again, after or during the storage of the lyo cake, compounds will start to evaporate. Typically, these are the volatile and semi-volatile compounds, and what you will see is that they will be adsorbed onto the lyo cake. Because of the fact that those compounds will be adsorbed, there will not be an equilibrium for those compounds between the gas phase and the rubber stopper. That means that there's a continuous release of those volatile compounds from the rubber stopper, and they will be accumulated onto the lyo cake. What you could see also is that some of those compounds potentially may be reactive, and that is more pronounced in lyo applications than it is in aqueous drug products or small volume parenteral aqueous applications. So you can see that the red dots here are the reactive leachables, and the other ones are the ones that have been absorbed onto the lyo cake. Now, the extent of that outgassing will depend, of course, upon the quality of your rubber or the grade of your rubber. Now we have looked at the primary packaging, now also the reconstitution of the lyophilized drug product will play a role. So what you typically do is you add the reconstitution solution either through a disposable syringe or through a pre-filled syringe, and then you start to shake that vial to solubilize the lyophilized drug product. What is happening initially is that all compounds that have been adsorbed onto the lyo cake solubilize, of course. That will mainly be volatile and semi-volatile compounds. However, there's also that short-term contact between the reconstituted drug product and the materials or the components. And that means that also non-volatile compounds and also potentially metal residues, some metal oxides, for instance, or ions, anions typically, may be released. So as a consequence, when we look at the techniques that we will use to characterize, for instance, the materials of construction in an extraction study, it will be a combination between focusing on the volatile and semi-volatile compounds with headspace, GCMS, and GCMS for the primary packaging of the Lyo applications. But also we will need to expand that into the non-volatile compounds, metals and ions, because of that short-term contact of the reconstituted drug product with the container closure system. So at the end, there's nothing really different than for any other small volume parenteral that is aqueous based from a extractable perspective. Now, taking one step back, as some kind of refresher, I would like to go through the general flow of an extraction study and identify the different steps here. In the first step, we try to discover all extractable compounds that are present on the materials of construction of the components that make up the container closure system. That means that with an orthogonal and complementary methodology, like I explained on the previous slide, that you want to establish a very detailed extraction profile, as detailed as possible. The question, of course, is how low should I go? And that is determined by, for instance, the AET level. Some basic information on the administration, on the toxicology, will determine how low you need to go. Typically, that is the analytical evaluation threshold that is specified in the PQRI, for instance, or associated toxicological thresholds. Once you have established how low you need to go, then everything that falls above that AET level needs to be identified, because it's only then that you can link the identity of the compound to its relevant toxicological information. We at Nelson Labs have published an ebook on good identification practices, and it explains the steps that we take in trying to identify compounds, whether they are extractable or leachable compounds. If you would be interested, you can download that from our website. And in the next step, we try to quantify those compounds, because it's not only the toxicity, but it's also the quantity of those compounds in the material that will play a role in terms of making that initial safety assessment. For the leachable study, you're looking at both a combination of long-term and accelerated condition. What the authorities always ask is the long-term stability, but if you would like to speed up your submission, it is possible to take the accelerated conditions and submit that first, as long as you then afterwards submit the long-term stability results also to the authorities. Of course, here also the AET plays a role. Everything that is above the associated AET for your drug product needs to be identified and quantified. You need to have different time points analyzed, because it is a strict requirement, for instance, for the FDA to look at the different time points. You need to look at the whole shelf life and check off across the shelf life what the trend is of your leachables being released and accumulated in the drug product. You're using more quantitative methods here. Typically, you validate your methods that can range from some kind of limited validation, a limit test, for instance, up to a full-blown, fully validated analytical method. And in combination with that, you typically screen also your drug product for unexpected leachables. And I will show you an example later on for that. For the administration devices, they typically have a transient or short-term contact with the drug. So that means that you do not need to test at every time point. You only need to include some kind of assessment. For instance, at the T0, these administration devices are typically more medical devices or combination products. Like I said, we will not address the the approaches here, what actually needs to be done in detail for those administration devices in this webinar. However, if you would be interested, you can always reach out to me. Now, coming to the challenges and consequences of the leachables profiling after reconstitution. The biggest challenge in setting up and designing leachable studies for lyo applications is to find the right blank. Now, before I explain what a blank is, this graph here, this picture, which is some kind of mirror image of a chromatogram, where the upright chromatogram displays the drug product or the analysis of the drug product that was in the packaging compared to the blank drug product, which had no contact with the container closure system, you can see that it's some kind of differential analysis. Every peak that shows up in the drug product that had contact with the packaging and that is not present in the blank will be the focus. It's some kind of comparative assessment, and it is a comparative assessment between the aged drug product, which is in the primary packaging, and the aged blank drug product, which underwent the same type of aging as some kind of baseline. Now, what is a good blank? And I have displayed here a fill and finish line. So a blank is a drug product that was not exposed to the primary packaging and is, as a consequence, is leachables free. So for a typical liquid application, a liquid small volume parenteral application, you sample the blank right before the liquid filling into the primary packaging, and you pull the samples that will have had contact with the container closure system right at the end of the filling line. The problem, as you all may know, is that lyophilized drug products or lyophilized or freeze-dried in their vial themselves. So there's no way you can pull in the same way the blank for lyophilized drug products as you would do for liquid small volume parenteral applications. So that means that you need to look for alternatives. This slide here is mainly to explain the value of a blank. This is analysis of a drug product, actually a lyophilized drug product, where we didn't have any blank. Here you can see a list of 75 organic compounds, which are potentially impurities combined with leachables. And if you now would like to know which ones are coming or are a result of the contact of the drug product with the container closure system, this is actually very difficult to determine here because you don't have any blank. You don't have any differential analysis. So that is really the challenge in pinpointing here which ones are the result of the contact of the primary packaging. So what are the alternatives? There's no true aged blank for the drug product in lyo applications. So what kind of possibilities do we see as some kind of alternative? The first thing you could do is say, okay, let's look at T0, the initial time point, and use that as some kind of blank baseline throughout the complete study. That is a possibility, but not a preferred option, because the problem is that some compounds may be already present after a short-term contact, and also the drug product didn't undergo any aging. So the risk here is that some of the degradation compounds of the drug product may be considered as leachables then. A second option is to use the upright position as the reference for the inverted position. But as we have seen, the interaction mechanism between the container closure system and the drug product is not really based upon the direct contact, solid-solid direct contact. It's an outgassing mechanism. So that means that this is really not an option at all. You could use lab-prepared or freshly prepared drug products. So that means that you take all the ingredients and you prepare some kind of blank that is solubilized already. This also has some disadvantages. Sometimes you can do that once, but then the stability of the drug product may be an issue over time. Or you could say, okay, for every time point that we want to analyze, we will do that preparation. Then you will have that less of a stability issue, but it remains an issue in terms of degradation compounds that may be missed. Another option is emptying the lyophile right after the manufacturing, but there the risk is that by opening the vial, some humidity comes in, and that the stability of the drug product will be affected in that way. So there's no perfect solution. It's also depends upon the type of drug product that will be studied. So as a consequence, for leachable studies for lyophilized drug products, you may need to rely more on targeted analysis and look at more quantitative methods. You should also add a screening step that you could do more via targeted way in looking to the extractable compounds that you have seen in your extraction study, for instance, and look if they come out, if these compounds come out. Let me. I'm sorry, my phone went off. Okay, so I'm sorry for that. So you need to add a screening step, and the screening really helps you to find those compounds that are not expected, and we will show you later on, in the next couple of slides, what kind of leachables could be detected, and we're mainly talking about those reactive leachables. So in conclusion, what you need to do in your leachable study is you need to evaluate trends. That means that you need to analyze time points across the shelf life. You need to be able to make a correlation between the results of an extraction study and a leachable study. This is mainly to verify if the design of your extraction study was okay and if you didn't miss anything. And you also need to evaluate the reactivity of certain leachable compounds with the drug products. Now, coming to the leachables side of the reactive leachables, what we have seen is that we see that there's a lot of reactivity, or the most cases where we see reactivity of leachables are in lyophilized drug product. And we see that with small molecule APIs, we see that with excipients or other ingredients, and we also see it with therapeutic proteins and peptides. And typically it are the electrophilic leachables that are reacting with the nucleophilic functional groups of the peptides and proteins. And the most occurring reactive leachables in the lyo applications are, like they are displayed here, are those brominated or chlorinated rubber oligomers, the C13 and C21s. A first example of an interaction that we saw is with the thiodiazole functional group of an active ingredient, where both the C13 and C21 reacted with that functional group. We also saw, for instance, an interaction with the C21 brominated oligomer with glycine, which is an excipient or a bulking agent for lyophilized drug products. We also saw it happening with an impurity of polysorbate 20, which is stearic or palmitic acid, where an adduct was formed between the C21 oligomer and the acid. So these are all things that can happen. If you would be interested in learning more about the reactive leachables and what kind of reactions could take place, I would like to refer to this article of the researchers of Amgen, which was published, I believe, in 2014 or 2015. And they explain what kind of reaction mechanisms can take place between the electrophilic leachables and the nucleophilic drug products. It's typically a nucleophilic substitution reaction, as in 2 or as in aryl, or a Michael addition, or a acylation or a shift base formation reaction. What we also did at Nelson Lab is based upon the rules and the predictions that were explained in that article to check on some of the most common leachables that we see, if actually that would take place. And we selected 6 here, and we allowed them to react or to be exposed to the lyophilized glargine, which is an insulin, a long-acting insulin, and it's a peptide. And actually what we saw was that we saw in all cases here an interaction between the peptide and the leachables that were predicted to be reactive. If you would like to find out more on this, there's also a webinar that you can consult, and you can find the references on the bottom of this slide here. Now, why is it so important to check on that? Because of potential immunogenicity concerns. And there's a good guidance of the FDA around that, and that's the immunogenicity assessment for therapeutic proteins. And there it is stated that leachable materials from the container closure system may be a source of materials that enhance immunogenicity, either by chemically modifying the therapeutic protein product or by having a direct immune adjuvant activity, including the following, and then they refer to the EPREX case. Now, what they also state is some kind of recommendations is that extractables and leachables should be performed to assess the attributes of the container closure system that could interact with and degrade protein therapeutic products. So you should do some kind of assessment to see if the interaction between leachables and the therapeutic proteins would not lead to any degradation of the therapeutic protein. Now, how to minimize the interaction between the rubber stopper, for instance, and the lyo cake? The only thing that you can do is select a higher quality of rubber. And as an example, I have displayed here again a chromatogram of an old formulation of a rubber. That is the upper side chromatogram. And then compare that to an extraction profile of a newer rubber. For people who are not familiar with chromatography and analytical chemistry, the number of peaks or the number of compounds that we detected and the height of the peaks or the surface area under the peaks is the concentration. So you can see for the old rubber formulations that you see a high number of peaks and they are large. For the more recently developed formulations, you can see that the number of peaks is substantially reduced and that the concentrations are also substantially lower. You could even go further and say, okay, what could a coating do? There are certainly coated rubbers on the market. Either they are film coated or spray coated. And that creates an extra barrier for the migration of those compounds from the rubber into the drug product. So it's certainly an improvement in terms of extractable and leachable risk of interaction. Now, also here, I'm trying to compare a coated rubber with the same rubber grade and compare that with an uncoated rubber. And you can see that even at the level of an extraction study, which is exhaustive or exaggerated compared to the exposure that you have in to the drug product, you can see that the number of compounds are substantially reduced because of the application of a rubber coating on film coating onto the or a spray coating onto the rubber material. So a coated rubber really helps in reducing the risk of migration. So reducing or avoiding the interaction is how could the selection of the rubber grade help you, and that is by taking clean rubbers and, if possible, consider coated rubbers, because then you can really minimize the interactions between the rubber stoppers or plungers and the drug product. Now, coming to the end of the presentation, let's look at the key learnings here of what we have addressed today. The first conclusion is that the regulatory expectations in terms of the need and the depth of performing extractable and leachable qualifications is ambiguous, and is actually based upon the expectation that it would be a solid-solid interaction between the rubber stopper and the lyocake, which is not the case. So the expectations of the authorities are different than what is written as a guidance. The interaction mechanism between the lyo drug product and the rubber stopper is not based upon a direct contact mechanism, but goes through a mechanism of outgassing. This may lead to some kind of accumulation of those volatile and semi-volatile compounds from the rubber onto the lyocake. And the adsorbed leachables may be reactive also, and may lead to the drug product degradation, and may affect the additionality, the safety and the quality of your drug product. You can only discover this if you add a screening step in your formal leachable study, and then you can evaluate the occurrence of the leachable reactivity. And this can assist in mitigating also the risk of the occurrence of immunogenicity through a chemical modification of the therapeutic protein, in case you're studying a protein, of course. And also consider the right rubber quality for your lyophilized container closure system. With the next two slides, I would like to refer to some of the work we have been publishing in the last 2 years in the PDA journal. And also here I'm making a reference again to the ebook that we have published on good identification practices for organic, extractable and leachables via mass spectrometry for people who would be interested. And the latest publications that were accepted by the PDA journal you can find on this slide. Of course, Nelson Labs, together with Sterigenics, has much more educational information, such as webinars that can be consulted, videos, seminars, white papers, and so on. If you would be further interested, please feel free to consult our joint websites to find the information that could be interesting for you. And with this, I would like to conclude my presentation and thank you for your attention and open the floor for questions.
Thank you, Peter. I think that was a really interesting and very comprehensive presentation. And you can see it by the number of questions that we got. So it's good that we have some time to tackle all of them. I just want to remind you that you can still submit your questions. We will try to tackle as much as possible in the time that we have before the end of the session. So I'm going to start reading a few of the questions, and please feel free to check if there is anyone that you really would like to tackle. Also, I will leave the presentation in this slide in the case that we don't have enough time. They can contact you if they wish to. But let's go with the first one. So there's a question saying, for extractables and leachable studies, do we need to perform it for entire shelf-life storage, considering that it is a continuous process?
Can you repeat that question, please?
For sure. So for extractables and leachable studies, do we need to perform it for entire shelf-life storage, considering that it is a continuous process?
Yes. So certainly the expectation of the FDA is that you show the trend of your leachables across the shelf life of your drug product. So actually you need to pull samples from your stability across the shelf life like you would do in a stability study. Maybe not all time points will be identical to the normal stability study, but across the shelf life of your drug product, you need to pull samples to study the leachable accumulation of leachable compounds in your drug product. That is an expectation and a requirement for FDA. And what we also see is, I mean, typically FDA and Europe are harmonized in that. So we always perform studies that show the accumulation of leachables across the shelf life.
All right. I'm going to continue with the next question. So there's another saying, for stoppers with special coating, such as Fluorotec and Omniflex, will the coating minimize the outgassing, leaching, extraction from the stopper body?
Yes. So because of that additional barrier, the fluor coating that has been put on top of the stopper, that really minimizes the interaction between the rubber and, I mean, it minimizes the outgassing of those volatile and semi-volatile compounds from the rubber stopper and being absorbed onto the Lyo cake. So that's would definitely be very helpful.
All right. Going to continue with the question. So there is another saying, what type of a spray coating is used that acts like a barrier? So I can repeat it. It's
yeah.
Yes. What type of a spray coating is used that acts like a barrier?
The type of spray coating, I understand. I'm not sure. I do not know if there are different types of spray coatings. I'm not really an expert in producing coated rubbers, to be honest. Maybe there are different types of spray coating, but I'm not aware that there would be more. And also, as a consequence, I don't know if there would be any difference.
All right, no problem. Let's go with the next question then. There's another question on, does all extractables and leachable compounds require toxicological assessment?
Well, there's a difference between extractables and leachables. Actually, what is required is that for all leachables that you detect and discover in your drug product, you need to do some kind of safety assessment. Safety assessment, there you need to look at two things. Look at the identity of the compound, because that relates to the toxicological information that you can find on the compound. And look at the concentration, because that will determine the dose of that compound to the patient. Of course, you can only say that a certain compound is not of risk if you have some supporting information on that compound. Of course, it is possible that for some compounds you should not make a full in-depth toxicological analysis, because it may be, for instance, a residual solvent that is of class 3, for instance, which can be in a drug product at 50 ppm. That is possible. You know, the level of effort can differ, but you will always need to find or make some kind of rationale to say why this compound that you found as a leachable in your drug product may not be of any concern.
Okay, I think that's a clear answer there. So I'm going to continue with another question that is sort of 2-in-1. So are those rubber oligomers that you always mention present in all rubbers? And as a second part, and are those rubber oligomers more of a concern for lyo drug products than for liquid formulations?
Yeah, well, they are always present in halobutyl rubbers, like the chlorobutyl and the bromobutyl rubbers. Of course, the concentration of those compounds in those materials may be different depending upon the grade of the rubber that is selected. There are other rubbers on the market that are not based upon the halobutyl or chlorobutyl rubbers. Those ones will not contain those types of oligomers. Of course, putting a coating on top of the rubber will reduce the interaction. So that means that those compounds, the C13 and the C21 oligomers, are volatile. Certainly the C13 is very volatile, so that will immediately evaporate from the material and will absorb onto the lyo cake if there's no barrier. And that is the same, but to a lesser extent for the C21. It's a semi-volatile compound, and the release of that compound from the rubber may go slower, but eventually it will be released and potentially also will be absorbed onto the lyo cake. And specifically for those compounds, the reactivity is really an issue. The reactivity is an issue, and the potential toxicity is an issue, because they are considered as alkylating agent, and that's what you actually see when they are interacting with the API or the ingredients or peptides or proteins. Of course, the question is how toxic are they? And that is something that is still under debate. We have done some Ames testing on those rubber compounds, so that information is available, but there's no publicly available information broader than that in the literature, I mean.
Okay. I'm going to go ahead because there is still a lot of questions. Probably you will have to tackle them afterwards. You will receive all the amount of questions, so you actually will be able to follow up. But there is another question on saying, well, a comment saying, because leachables do not come from direct contact between the rubber and the lyo product, would you recommend performing a stability testing in inverted, upright position, or both?
We have done studies where we inverted the lyo vial and upright. We didn't see any difference. For that particular study, or those cases where we both studied the inverted position and the upright position, we didn't see any difference whatsoever. So we don't see that because of the contact of the lyo cake with the rubber, that there would be an enhancement of the mass transfer from the rubber to the lyo cake.
All right. I'm going to go ahead to ask you, I think, the last couple of questions, I think, because we have maybe time for that. So are abducts that are being formed as a result of reactive leachables considered as leachable or drug impurities?
That's a difficult question, and the problem is there's no harmonization also within the authorities. We have seen cases where those adducts between, for instance, an API and a reactive leachable was considered as a drug impurity, and that was then ICHQ3B was referred to, and in ICHQ3B impurities of the API are monitored at a substantially higher concentration compared to leachables. However, we have seen the contrary also, where they are considered as leachables. We also saw cases where not only the API but also a degradant of the API was reacting with the oligomers, for instance, where a whole investigation needed to be done to prove that those adducts were not harmful. So there's no harmonized approach or no guidance at this moment, from my perspective, to say, okay, it's drug product impurity governed by the ICHQ3B, or it is a leachable. So for me, the situation at this moment is not really clear.
All right. So I think I'm gonna probably ask you this last question, and then feel free to give your final remarks too here. So this is the question: should the leachable assessment of the drug delivery device be included into the leachable assessment of each time point in a leachable study?
No, like I said, of course, those drug delivery devices or administration devices, they can really contribute to the impurities profile or the leachables profile of the drug product that will be eventually administered to a patient. That is clear. Only it's a transient contact. It's not something, those delivery devices are not in permanent contact, only during those few minutes or few hours that the drug will be administered to the patient. So that is certainly a phase that will need to be checked off to see what is the contribution if, for instance, if a drug product is flowing through an administration tube before and after it flows through that administration tube, what is the difference in leachables profile? But you don't need to do that every time for every time point, because it's if you do that once, that's okay, because then you know, if you do it once, what kind of contribution it will give.
All right. I think I just want to close basically saying that you were happy to share your slides. You can find them, if people want to download it, they can do so in the related content tab. So please do so if you really want to check them again. I think now the slides is on your contact, so they can also contact you in case they have any questions. You will be able also to follow up with the people that attended and submitted questions that we were not able to tackle today. And basically what I wanted to say is thank you for taking part in this session. I think you have given a really nice and immersive experience into the leachables and extractables. And I just wanted to say also thank you to our sponsor, Nelson Lab, for sponsoring this session. And also a big thank you to you, our audience, for your attention.
Okay. Thank you very much. So if there would be any open question, maybe the questions can be sent to me, and I will be glad to answer those questions to people who have asked them.